Hello everyone, this is Devobani Ganguly. I welcome you all to the annual Distinguished Lecture Series 2021 organized by our Center for Health Science and Technology at JIS Institute of Advanced Studies and Research, Kolkata. It is an opportune moment to start this lecture series as a part of the celebration of the second Foundation Day of our institute. During this entire series, we will have the opportunity to learn from highly distinguished scientists from India and abroad on the diverse aspect of medical biotechnology and bioinformatics research and its diverse applications to mankind. I believe all of you have already received the abstract brochure and browsed the 6 abstracts of the lectures and equally excited to learn from the talks as we are. The brochure also contains information about our institute and the courses we are running. If you are interested to know more, mail us or call us. All contact information are given there and also visible on screen. It gives me immense warmth and great pleasure to grace all of your presence today in the first lecture of the series by Professor Daniel Huson. Thank you so much, Professor Huson, for accepting our invitation. Live streaming of this talk is available on our YouTube channel, CHIST, JISIASR. Please write your questions to the chat section of Zoom or comment section of YouTube. Now, may I request Dr. Shandip Paul to introduce our speaker. Thank you so much, Dr.
Thank you, Devobani. So it is my really great privilege to introduce today's speaker, Professor Daniel Huson. Professor Daniel Huson studied mathematics at the University of Bielefeld in Germany, where he received a PhD in 1990. He was a postdoc at Princeton University and the University of Pennsylvania from 97 to 99. And then he joined Cellular Genomics Corporation, where he worked on the assembly of human genome. He has been professor for algorithms at the University of Tübingen since 2002. He was a visiting professor at the Nanyang Technological University of Singapore from 2011 to 15, and at the National University of Singapore 2015 to 20. In 2020, he was awarded an International Leader Fellowship of the Royal Society of New Zealand. His major research interests are the development of algorithms and software for applications in phylogenetics and microbiome analysis. He has developed several state-of-the-art sequence analysis softwares like Splittree, Megan, Diamond, to name a few. So we used to use all these things from beginning of our research career. So these algorithms have actually transformed the traditional sequence analysis method. So we are today fortunate to have him with us. And today he will be talking about microbial phylogenetic context using phylogenetic outlines. So without further delay, I am welcoming Professor Huson. Now over to you. Thank you so much.
Yeah, thank you very much. What am I trying to do here? Start share. Okay. Let me try and share my screen. That's always the hardest part. Is that good? Oh.
Yes.
Okay, thank you very much for the kind invitation. It's a great honor to give this presentation. And so I'm going to talk about phylogenetic context and phylogenetic outlines. And this is joint work with Dave Bryant, who's at Otago University in New Zealand, and with a PhD student, Jana, here in Tübingen, and a other student, Banu, who's in Turkey. So here's the outline of the presentation. So each chapter is a standalone chapter, but hopefully it'll all come together towards the end of the talk. So I'm going to talk about a number of different threads and hopefully bring them all together in a meaningful way. So I'd like to start with phylogenetic outlines. So in phylogenetic analysis, we like to use phylogenetic trees to represent the relationship between different species or different organisms, different taxa. And there's many ways to compute phylogenetic trees. And one of the ways that you can get to a tree is by computing a distance matrix and then using the neighbor joining algorithm. This is a widely used method, and the distance matrix sometimes comes from an alignment, a multiple alignment of sequences, but there's other ways to get distance matrices, for example, based on some K-mer content analyses, and we'll get back to that in a later part of the talk. So, but the basic idea is you compute distances between your taxa, and then you visualize those distances using a tree. And thing about a tree, though, if we look at this for a moment, so these are bacterial genomes, and as we all know, bacteria share a lot of genes. There's a lot of so-called horizontal gene transfer, and so it's questionable whether you can always represent the true relationships, but evolutionary relationships using a tree. And so sometimes people consider using a phylogenetic network to represent the relationships between different bacteria. And here's an example of one of those networks. So this is again using a distance matrix, and then rather than using the neighbor joining algorithm that produces a tree, you can use something called neighbornet, which computes one of these networks. And I'll talk a little bit more about these networks, give you some idea of in which way they generalize the concept of trees. So actually, it's a two-stage process. The neighbornet computes a set of what are called splits, and then there's another algorithm that you then apply to that set of splits to actually get the full network. And so this talk today is introducing a new type of network that we call a phylogenetic outline. So this is something that David Bryant and I came up with, and so this will be one of the focuses of this talk. And so let me dig into that a little bit deeper. So this is a phylogenetic tree, as we already discussed. And one of the nice things about a phylogenetic tree from the computational or the visualization side is that it has linear size. So if you have n taxa, n organisms that you want to put in the tree, well, you have n on the order of n edges and nodes. So a tree itself is a very lean visualization tool. And it's easy to phylogenetic tree. We like them because they're easy to interpret. We think of speciation events at the internal nodes and so on. Some of the downsides of a phylogenetic tree is they're not so good at showing uncertainty. And they don't, by definition, they don't show alternatives. So phylogenetic tree, once you've drawn it, you've decided the order in which things have evolved, or speciation order, and it doesn't show alternatives. Obviously, people have come up with many ways to annotate trees and to show some of this information, but a phylogenetic tree itself doesn't do that for you. Whereas one of these split networks, it does show uncertainty. So if you look down here in this green circle, you can see, okay, it's not quite clear how these different species branch away from each other here. And it does show alternatives. So this what looks like a big, this big diamond-shaped thing here, not diamond's not the right rectangle, not rectangle, this big open box here shows that on the one hand, there is support for this edge, which also appears in this tree, separating all these taxa from everybody else, but there's also nearly the same amount of support for an alternative taxonomy of phylogeny where these guys get grouped together. Okay, so one of these networks can show alternatives, multiple trees simultaneously, if there's support for those multiple trees in the data set. Downsides of these split networks is one is they are large. They have n to the 4 edges and nodes in them. So that's quite a gain in complexity. And they do, sometimes they don't look so complex because many of the edges are parallel edges like this, and it just looks like a fattened up edge. But they are sometimes confusing to interpret. Biologists like to look at individual nodes in one of these networks and say, okay, what's going on here? Is it a speciation event in multiple directions and so on? So that's kind of confusing. And so we come up with these outlines that look like this. They look very much like the split networks, except you don't see all those details in the inside. So they can show uncertainty, so you still have a little bit of uncertainty visualized here, and they still maintain that ability to show indicate alternative groupings. But in terms of nodes and edges and so on, they only have quadratic size, and they're easier to interpret than those split networks. So this is a new visualization of phylogeny that we've come up with called phylogenetic outlines. So that's like the first part of this talk. And what I'm going to do, hopefully, is show you that these outlines have a good use when you're trying to place new genomes that you've obtained into the context of known genomes. Okay, so, and one thing just to add that to this talk, most methods, phylogenetic methods, produce unrooted trees, but in the end of the day, if we're really trying to interpret what's going on in terms of evolution, we do need to root the trees. So we have a direction, a time axis, and one way that we like to do that, for example, is using an outgroup. So there's this Contendobacter here, which is the furthest away from all these other Accumulobacter. So we can use that as an outgroup, and then we can redraw the tree in a rooted style. And you can do the same with these outlines. So here's a phylogenetic outline for the same data. Here's our outgroup again. And so we've implemented algorithms that allow you to root your outline by outgroup or by midpoint routing. Okay, so that's a new visualization technique that takes as input a distance matrix, and it provides us output a visualization that's more general than a tree, but not as messy as a split network. Okay, so we'll get back to those towards the end of the talk. Now I wanted to talk about some more general things, and then hopefully it'll all come together at the end. So phylogenetic is a placement that phylogenetic placement is a problem that a lot of people worked on. For example, Eric Mattson, more than 10 years ago, or he came up with the p-placer algorithm that's widely used. And here the computational problem is you have pre-computed a reference phylogeny, and now you've got some new query sequences, and you want to find where those queries fit into the reference. So what's the best place to put them? So this is a picture indicating that problem. So you've computed this reference tree. You spent a lot of time coming up with a tree that you believe in, that you think is a good representation of the true phylogeny. And now you've got some sequences, and you want to know where's the best place to put these. And so you use some maximum likelihood criterion or parsimony criterion to find the best place to place those things. And you get something like this, where these three query sequences have been placed somewhere into the phylogeny. So that's an important problem, and algorithms for that are widely used. Find the best placement in a pre-computed reference tree. And we will be talking in a moment about GTDB, the GTDB Toolkit, in more detail. And it provides, one of the things it provides for you is that kind of service where you have your newly assembled genome, microbial genome, prokaryotic genome, and you want to know where it fits in the big scheme of all the other reference genomes that people have already produced. And so you might be using phylogenetic placement to find the best placement for your reference sequence. Okay, so let me talk more about GTDB and GTDB Toolkit. So this is work that we've not been involved in. This is not our work, but I think it's really important work, so I like to describe it nevertheless. So the GTDB is an attempt to provide a similarity-based taxonomy and database for prokaryotic genomes for applications in microbiome analysis. So quite often people use the NCBI taxonomy for microbiome analysis. But the thing about the NCBI taxonomy, its main goal really is to accommodate all the sequences that are present at NCBI. And it doesn't really always follow sequence similarity and evolutionary relatedness as closely as you would want it to. So, but GTDB is an attempt to provide a taxonomy that's really based on similarity of certain marker genes and DNA similarity. And so the GTDB taxonomy, or the GTDB database is based on 32,000 so-called representative genomes, and the extended version contains 195,000 genomes. Many of these are MAGs, metagenome assembled genomes, which are important. If you do a metagenome analysis, you want to use as much current data as you can to perform that analysis. So the GTDB database and taxonomy, they're developed expressly for microbiome sequence analysis, and the GTDB taxonomy provides an alternative to the NCBI taxonomy. And so in a tool like Megan, one of the tools that we've developed, this is a tool for doing interactive metagenome analysis. For your data set, you can open up a taxonomic analysis of your data set based on the NCBI taxonomy. And here you can see, going from left to right, here's a big bacterial node, and underneath those bacterial nodes, there's multiple phylum level nodes, and the size of the nodes indicate how many of your reads have been assigned to these different nodes. So this is all based on NCBI, but you can also look at your same data through the eyes of the GTDB taxonomy, and then you can see there's a lot of similarity. Obviously, the NCBI taxonomy is a good taxonomy, but this is just a refined taxonomy. So in details, you will see that GTDB might bin your data slightly differently. Okay, so GTDB is an alternative to the NCBI taxonomy. And the GTDB Toolkit provides tools for analyzing your data. So I'm going to describe those, and then later I'm going to describe our approach, which is an alternative to the GTDB Toolkit, using those phylogenetic outlines that I mentioned at the beginning. So the GTDB Toolkit provides some methods for you. It provides methods or tools for doing phylogenetic placement. So you've got some genomes that you've obtained from a metagenome, and you want to know where they fit in to the taxonomy of reference genomes. And so it uses that pplacer program that I mentioned at the beginning, that likelihood-based taxonomic or phylogenetic placement program. So you can do phylogenetic placement of your query genomes, but it also provides a tool for doing de novo phylogenetic tree inference, where it throws your genome into an analysis with representative genomes. Okay, and so this is what the output of such an analysis looks like. So here's an assembled genome, and I'll talk more about this later. And this is the typical output of the GTDB toolkit when you do phylogenetic placement. It says this is where your query sequence is, and these are the reference genomes that are close by, and they're all in the same genus here of Torera. And here's an example of a de novo analysis of that same data. It looks very similar. You have reference genomes nearby. They're all in the same genus of Torera again. And then you have these numbers here. These are bootstrap values or confidence values of how confident these groups are. Okay, so the aim of this talk here is to show how we can use those phylogenetic outlines that I described at the beginning to display what we call the phylogenetic context of some query genomes. So where does your genome that you've produced lie in comparison to known genomes? And I mentioned at the beginning, one way to compute trees or these phylogenetic networks is to first compute the distance matrix. And so there's been a lot of really nice work done that aims at using K-mer sketches to represent genomes to allow rapid comparison of large numbers of genomes. So we're going to talk about MASH distances as an example of that. So in the context of modern biology, when we're looking at microbes, we know that there's on the order of 200,000 genome sequences. And if you want to be able to compare those, and each, on the order of multiple megabases in size, if you want to be able to compare those, you need very fast techniques for comparing reference genomes. And so MASH is an example of a whole range of methods that all basically do two steps. They take the genomes that you would like to compare, and they extract all kmers, all words of length k from those genomes, and then they don't keep all the kmers, or they're extracted in some way or another. And the methods, there's different methods to do this in different ways. They maintain a subset of those kmers, which is called a sketch. And then to compare two genomes, you compare their sketches. So each genome comes with a small sketch, and then to compare genomes, you compare their sketches. And it's based around the idea of looking at the kmer content and then doing basically what's called the Jaccard index. Which kmers do they share? How many kmers do they share, divided by the number of kmers that are present in total. Yeah, so I'm not going to talk too much more about that. So that's a basic approach. MASH is probably the most well-known of those approaches, but there's a number of other similar approaches. And there's quite a lot of research going on in that area at the moment. People are trying to improve this, have smaller sketches that are faster compute and are more accurate. In any case, these kind of sketches give us a nice way to obtain distances between genomes in large numbers of genomes. And so one thing that you can think about doing is, okay, if I want to place I've got my query genome, I've just assembled a genome, I've extracted it from a microbiome sample, and I want to see where it lies in comparison to all the known reference genomes. Well, the way to go would be to compute its MASH sketch and then compare the sketch of your query against all the MASH sketches for all the references. And the way that people like to do that, one way to do that is to use what's called a Bloomtree. So now, at the moment, this is a bit technical, but hopefully we'll see that it's useful. So the idea is you've got the GTDB taxonomy describing all the prokaryotic genomes. And then at the leaves of the taxonomy, you have the individual reference genomes. And for each of those, you compute a sketch, one of those MASH sketches. So a MASH sketch is usually a set of kmers, say 10,000 kmers of length, 21 each. Okay, so you have all these, well, here, in fact, I said 1,000, yeah, but something like this. Actually, for our implementations, we use a sketch size of 10,000. So for each genome is represented by 1,000 or 10,000 short words of length 21. And your query comes in and you want to quickly find which reference genomes share have a non-empty intersection with the sketch of your query. And so what you want to do is you want to do pre-processing on that taxonomy to make that an easy and fast query. And so what you want to do is for each internal node of the taxonomy, you want to keep the set of all kmers that appear below it. Okay, so each node knows which kmers are below it, and then the idea is you've got your query, you extract the kmers in your query, and then you compare against a node, the root node, and then you ask the root node, okay, which of your children contains one of these kmers from the query, and say, oh, okay, this one contains one of the kmers, and then you do the same thing again. Now, which of the children contain one of these kmers, in this case, this guy. And so that way you can navigate through the taxonomy from the root down to only those leaves that have a non-empty intersection, where the sketches have a non-empty intersection. And that way you can quickly figure out to which genomes your genome has a significant similarity. And the technical thing that people like to do here is, rather than just using a full list of all the kmers that appear below a node, you use a data structure called a Bloom filter, which is a representation of a set of kmers that has a fixed size. And the thing is that fixed size comes at a price that sometimes when you ask a set, do you contain this kmer, it's going to give you a wrong answer. It's going to say yes when the answer really should be no. So sometimes you might go down a part of the tree where you end up with nothing. So you're never going to miss anything, but you might do a bit of extra work just because of that trade-off between size and correctness. Okay, so we've computed sketches for the whole of the GTDB taxonomy and the Bloom filters for all of the internal nodes, and we provide these as an SQLite database with these tables here. And using a kmer size of 21 and a sketch size of 10,000, this database is about 6.6 gigabytes in size. This database is then what you would use to figure out the phylogenetic context of some genomes. So that's what I'm going to talk about next. So far we saw that method of phylogenetic outlines. You've probably forgotten what that was now, but we will come back in a moment. And then we saw that an important question in microbiome analysis or in any kind of analysis where you're looking at prokaryotic genomes is how can I figure out which reference genomes is my new genome, which ones is it similar to? Yeah, okay, now we're gonna. So we are trying to introduce a new word here. So phylogenetic placement was that problem where you place a query into a given reference tree, and phylogenetic context. We got a query genome, and we want to know basically what is the context of this genome? What are the related reference genomes? And so here's an example. So the examples are all taken from a paper that we published in 2019, where we did nanopore sequencing of a bioreactor sample, and we got some very nice high quality and medium quality draft genomes out of that sample. And so here, for example, BA is from that paper. It's a metagenome assembled genome consisting of 25 contigs of size 4.6. So on the typical size of a bacterial genome. Medium quality draft genome is meaning that it doesn't look like it's a complete genome, but based on certain marker genes. And now, so this is what we're calling the phylogenetic context. You can see here this metagenomically assembled genome, and we've computed a context. And you can, so we're using one of those phylogenetic outlines, and then you can see all these reference genomes here that come from the GTDB database, and that were found using that approach that I just mentioned, where we compute a MASH sketch for this query, and then we compare that MASH sketch against the MASH sketches of all the reference genomes using that Bloom filter tree. And then this is what we're calling in phylogenetic context. We get a good idea. Well, it looks from this analysis here, I'm guessing that it's a Tauera species, because everything that I see here, well, nearly everything, there's something different over here. We got proteobacteria, something or other, but everything else is a Tauera. So this is probably going to be a Tauera. And so this is implemented in SplitStree 5 program that we developed for phylogenetic networks. And so what you do to use this is you provide a file containing the context that you want to place, or for which you want to compute the context. And then you set up, there's some ways to set up what labels to use. Turns out that labeling entities in a plot is quite an important and sometimes tedious problem. So there is support for coming up with nice labels. But once you've done that, you basically press on this button here, and now this is how long it takes to compute the context. So I've got my 4 megabases of sequences, and I've just asked SplitStree to figure out what the context is. And you can see it's doing the search, it's using that Bloomtree, and now it's found everything. It's figured out what the distances are. So it's found 6,000 genomes that share at least one kmer with my query, and then you can see the spread here over the distance, MASH distance. So it estimates the distance between your query and all the reference genomes, and you can see that there's quite a spread there. And then as a user, you would then set a threshold. You say, okay, I want everything up to a certain distance, and then so here I'm setting a threshold of 0.3. No, I guess I'm not. No, I'm setting this. Here's the threshold here. Oh yeah. This is a threshold here of 0.18 or something, and then there's 31 genomes that are that close to your query, and then you can see a visualization of those 31 genomes together with your query. So it gives you a very quick and interactive way to place new sequences. And usually in a query file, you will have multiple entries or multiple contigs, and so quite often you want to treat the whole file as one entity, as we've done here. So the whole file of all those contigs has been placed or in the context of these other genomes, but you can also run interpret each of the FASTA records as a separate entity, and then you get a visualization that looks like this, where some of the larger contigs are close to the reference genomes, but then you've got a bunch of other contigs. Basically, the higher the number is here at the end, the smaller the contig is. As you can see, as the contigs get smaller and smaller, they're just more and more distance from everything else. At some point, it makes no sense to actually include these guys. Yeah. Okay, so there's two different things here. There's placement and there's context. So let's go back to GTDB Toolkit. It provides phylogenetic placement, so you can run the GTDB Toolkit for your query. So here in this case, it's a single contig, 5.2 megabases. It's classified as a high quality draft genome. That comes from that paper that I mentioned, where we did some nanopore sequencing on a bioreactor sample. So if we run that through GTDB, we get this output here. If we run it through SplitStree 5, we get this visualization here. And what we've done, yeah, so that's, and the third thing you can do is, so you can do phylogenetic placement to get this readout. You can do phylogenetic context to get this readout, or in GTDB, you can do a de novo analysis to get this readout here. And so how do these different analyses compare? So in terms of placement, one thing that's a bit inconvenient with the GTDB toolkit, but is that they don't provide the NCBI names for you. They provide these accessions that they've assigned. And so in our code, we provide both the accessions and the NCBI names so that you've got a better idea of what's going on. So, but to make it easy to compare the results here, we're using this color coding. So you can see there's a sibling in the placement analysis between B2, and it has this dark blue sibling here. And if you look over here, here's that dark blue sibling. They're right next to each other in the SplitStree 5. And if you analysis, and if you go further out, then you've got these guys over here, which also appear over here. I guess one of the colors is missing. And so if you do a careful comparison, you do see that taxonomic placement, de novo analysis, and phylogenetic context do provide similar placements of the genomes. Not always exactly the same in the details, and it also depends on whether they're, what the reference situation is. If you sequence a genome where there's no close references, then all of these methods are going to do worse in different ways. But yeah. Selling point for the SplitStree 5 approach is, if you're a biologist and all you know how to do is to click on buttons in the program, then SplitStree 5 is a completely interactive program. For the GTDB toolkit and so on, this is all command line driven. So you have to run command line programs, then you have to get some external tree visualization tool to do the visualization for you. So these methods, I guess, could be improved in terms of usability. They're not on the same level as this method over here, where you really just load your data and then wait for the analysis to conclude with a visualization. Yeah, and I think using a phylogenetic outline rather than a tree is probably a more appropriate way to go, simply because, as we discussed at the beginning, a tree, once you've decided on the branching pattern, for the person who's looking at the tree, they really don't see whether this is, how solid is this branching pattern? Is there an alternative that's quite close that is just not shown? Yeah, so you might see that more potentially better with an outline rather than a tree. Here's another example. This is from that same paper. Now it's medium quality draft genome with that B8. And then if you look at it in the placement analysis, you see, and then compare that to the phylogenetic context, you again see this similar pattern, that there's a close relative in both. And then there's purple further afield, these two guys. And then even further afield, the two red siblings. And then as you go further out, you find all these guys. So in this particular analysis, both methods methods find exactly the same context or placement. Yeah. Here's a third example. This is one of the so-called low-quality draft genomes where they're not, the draft genome, low quality here means it's not particularly complete, it's less than 50% complete. And here you can see that there's more discrepancy between the placement analysis and the context analysis. So the query sequences down here has this Nitrospira sibling. So the two analyses agree who's the closest sibling, and then there's this group of genomes over here, which are also reasonably close to these guys over here, different Nitrospira species. And then some of the genomes that appear here also appear here, but there's also a lot of stuff that appears here that doesn't appear here, and vice versa. So they're not phylogenetic placement and the phylogenetic context are not the same analysis, so they don't produce exactly the same results. But in both cases you do get quickly a useful indication of what are the related reference genomes for my newly sequenced genome that I know nothing about. Gives you a good place to start when you're trying to figure out, okay, what is B13 in terms of phylogeny? Well, it looks like it's Nitrospira, and it looks like it's more closely related to this species, it's less closely related to this species, these species, and these even further afield. You get the same view over here. So where you'd also expect phylogenetic outlines to be useful is in the context of strain level analysis. Well, strain level analysis, I think we all agree there that a tree is quite a questionable way to visualize data. So here we've been looking at trying to do a strain level analysis, for example, for Microbacterium tuberculosis. Here is a different isolates from different humans. There's one animal pathogen, so that's a nice outgroup to everything else. Everything else is from humans. And if we look at this guy here, for example, I've forgotten why we're looking at this guy actually. This guy looks from this analysis. This looks like this might be a hybrid. It might be a horizontal. Yeah, this might be on the one hand related to all these Russian strains over here, but there's also strong signal separating this from the UK. Yeah, so what I'm trying to say is these Russian strains on the one hand have a similarity with this UK strain on the one hand, but on the other hand with these other strains, these India and Italian strains. So potentially a strain level analysis might be able to give you a more informative picture than just a tree that would have to decide, am I going to put this Russian strain with the UK strain, I'm going to put it with these strains over here. I can't have it both ways in the tree, but I can have it both ways in the outline. Yeah. Okay, so I guess that was quite a short presentation. I hope that's, yeah. So I started out by talking about phylogenetic outlines, and then I hope I could convince you that a phylogenetic context is an alternative to placement. It's not going to replace placement, but it does something along similar lines in a slightly different way. We saw, well, I hope I could convince you that phylogenetic context can be computed quickly using MASH distances for the basic comparison, but then using a Bloomtree to quickly find the references with similar MASH with a good MASH distance. Phylogenetic outlines can display ambiguities in context. And this is all implemented in SPITS-Tree 5, and it provides an easy and fast approach to computing outlines for prokaryotic sequences. And yeah, these are the people I'd like to thank, and thank the Royal Society of New Zealand who funded part of this research. And I'd like to thank you, everybody, for listening to this talk.
Thank you, Daniel, for delivering such an illuminating talk. I mean, I can understand how important this phylogenetic context is in terms of placing contexting the draft genomes in case of microbiomes.
Yeah.
I mean, from either from soil microbiome or either from the human microbiome or any kind of microbiome, how important it is for different aspects, placing, I mean, contexting the proper space in phylogeny. That's the first thing we do. I mean, then we go for
analysis. First question, isn't it? What's the identity of this particular genome? You want to know that. You want to give it a name.
Yes, exactly. So the session is open for questions. So please, I mean, in the chat section, you can write questions, and we are happy to take questions. I mean, so I have, let's start. I mean, I have one question actually. So, I mean, as I understood that using this phylogenetic context, we can easily identify the closeness in terms of the horizontal gene transfer or the genomic islands. I mean, which we generally miss in case of GTDB and other types of phylogenetic placement. Say two genomes, I mean, both having the same kind of horizontal gene transfer happened, similar kind of genomic islands they got from the environment. So those things will also be included in case of phylogenetic context.
Yeah, if they lead to, So if there's, a phylogenetic tree is assuming that the only similarity is going to be through vertical transmission. If there's a lot of similarity due to horizontal transmission, that's usually lost in the tree because the vertical signal is always going to be the stronger one. But with the outline, in theory at least, if there's a strong horizontal, if there's a lot of horizontal transfer being going on between two otherwise quite distant genomes, in theory you should be able, you might be able to see that.
Yeah.
There's no guarantee that you will, but at least in theory you could, if the signal is strong enough.
Exactly.
Yeah.
And I think, I mean, I don't know, but I'm thinking about that. So I got a question actually. So thank you so much, Daniel, for such an insightful talk. A couple of questions, though not from the metagenomic perspective. Number one, your phylogenetic placement approach seems ideal for placing the continuously sequenced genomes of SARS-CoV-2 isolates as query in the pre-computed phylogeny. Is this being applied for this purpose? How deep can we go there to identify the new variants?
Yeah, I'm not sure that these kind of approaches are going to be are the right kind of approaches for identifying new variants, because new variants, I guess, there you really are looking at individual bases, individual mutations that change the configuration of the spike protein or something that change. And so these kind of methods, these KMA methods, they're usually aimed at, a broader, bulk similarity rather than a specific modification. So I guess if you're trying to identify new variants of a virus or so on, at the end of the day, you are going to use alignment. So this is all alignment free, and you do a careful alignment, and you see, okay, now where's the amino acid change in a protein or something. So I don't think, these kind of techniques are not, that's not their main strength. No, I would,
So second question is, I'd like to know if the phylogenetic outline approach could be useful in distinguishing mutational changes from recombination events in rapidly evolving species, such as Helicobacter pyrrole, Neisseria, or Streptococcus.
I mean, what you would hope is that you'll be able to see them. Let me see if I've got a picture where, not that one, not the strain, number one. Yeah, none of these really have so if there is a lot of recombination going on, I'm not claiming that there's a lot of recombination going on here, but in this kind of, let me just put that back into, whenever you see this kind of thing, a little triangle where this is indicating 2 ways to place something. This is maybe over here, this is slightly stronger, but still quite a weak one. This is saying that, okay, you might be, there's evidence saying that you group with these guys, but there's also evidence saying that you group with these guys. So if this triangle sticking out was a bit longer, then that would be a visual indication that there might be recombination going on between those three groups. So if there is recombination going on, then you do sometimes see that in terms of these triangular configurations appearing. Yeah.
So thank you. So another question from Raghavendra. Thank you, Daniel. Can phylogenetic outline be used for set of protein sequences that share similarity in the twilight zone? Will it benefit such sequences, or will the ambiguity be too much for any meaningful interpretation? Your thoughts, please.
So I'm not sure that I caught the whole of that. Is that written on the chat, or?
Yeah, from the chat. Yeah, I'm repeating. Just can phylogenetic outline be used for set of protein sequences that share similarity in the twilight zone?
Okay. Yeah. Now I understand. Thanks. So these are proteins that have very low similarity, just on the borderline to being random. Yeah, you can't. Yeah. Yeah, I'm not sure. Okay, so the outlines themselves, rather than, no, I don't think so. I don't. Yeah, I'm not quite. Yeah, the outline. So the outline is when you've got. So this sounds more like a situation where you have little or no phylogenetic signal rather than ambiguous signal. So phylogenetic outlines are more useful when there's multiple sources of phylogenetic signal, contradictory sources. Like on the one hand, there's a strong vertical signal through descent, but on the other hand, there's a lot of horizontal transfer going on. So you've got two incompatible signals, and then a tree building method is going to pick up one of those signals and say, okay, this is what it looks like. Everything else is going to be hidden. Whereas an outline potentially could say, okay, there's the main signal is these guys go together, but there's actually another signal saying, no, these guys go together. So ambiguous signals but strong signals will be picked up more likely by an outline, whereas what's described here is weak signals and probably not.
Okay. So another question is, thank you, Daniel, for such a beautiful lecture. Can the approach of phylogeny be used to identify gene families associated with higher prevalence in patients with a particular disease condition? So I think,
identify certain gene families, so that, yeah. So what I've presented here, this is all really genome based. So we're looking at, where does a genome fit in the context of the reference genomes, and where this question is asking about individual gene families and if you go through different samples, whether that gene family, you're trying to correlate gene families with disease state. That's a different question. I don't think this is necessarily, at least not what I've described. Isn't that helpful for that question, unfortunately, although it's an important question, obviously.
Yeah. So I have just a small query, like if some user want to use their own kind of database of different genomes, so is it possible? Utilizing
that is possible. Yeah, that is very possible. Yeah, that is possible because so we've documented, so that's a nice thing. Yeah, so we provide an SQLite database. So it has has a documented schema, and there's just a way of naming things. As long as you follow that schema, the split tree is not going to know whether it's the database that we provided or it's a database that the user provided themselves. Yeah, so it's flexible in that. So we are thinking about looking at other applications. For example, chloroplasts. That's a database of chloroplasts, because chloroplast genomes they're obviously not as big as bacterial genomes, but big enough for a MASH approach to work here.
So why I asked this question, because I was thinking that in case of genome scale metabolic models, suppose we have some reference genomes and we know their genome scale metabolic models. Now I find a new genome and I want to place that genome, I mean, using the phylogenetic outline to the nearby kind of.
That would be easy. That would be very easy to do. So we do especially. So what you need, okay, you've got your original genomes, and you have to compute the MASH sketches and the Bloom filters.
Okay.
And we provide command line programs for that. So you can compute all those and then put them into the database. So
okay. Yeah, that would be very much helpful actually. I mean, the first thing if we get a new genome, say in OTU, we find a new genome from the OTU file itself. So first thing is how to place. I mean, understanding its context, and then we can actually go for the modeling and other things. So there are many. I mean, thank yous. I mean, for the talk, and it's a really great pleasure. I mean, for this nice talk, and thank you so much. And on behalf
it's a pleasure.
On behalf of the chaste JISR family, I convey my sincere gratitude for your time, and we hope to get you in near future with this kind of
thank you very much. Thank you very much for your interest.
So I'm ending this session. Thank you all.
Goodbye.
Bye.